Schmiedel, talking about determining protein structures using deep mutational scanning.
All right, you can hear me. Well, wonderful. All right, let's jump right in. So I guess most of you, also me, by training, are kind of used to think about proteins from a physical chemical perspective, right? It's an ensemble of a linear chain of amino acids that folds higher order structures by physical forces, right? They hold together by physical forces. And also, most we know about protein structures, and all of the protein structures we have solved, actually come from the application of physical methods to these problems. So X-ray crystallography, NMR, cryo-EM more recently. However, I hope that most of you can also sympathize with this kind of genetic perspective of protein structures that I've put up on here, which is that the structure of a protein is actually central to the genotype-phenotype relationship in protein coding genes. And that is, we have a coding sequence that determines the structure of the protein, and the structure in turn determines the function, right? So the structure is really kind of the transforming functions that turns the genotype into the final phenotype that can be selected on. And my lab has been thinking about how genetic variation maps to phenotypic variation and the mechanisms behind that for a long time. And so here, what we've been asking is the question that, well, if we have a lot of genetic variation of a protein of interest, and we were able to comprehensively read out the phenotypic effects of each of these variants, shouldn't we be able to detect patterns and relationships that can somehow tell us what the underlying protein structure is, like, and how it has transformed genotype to phenotype? Right? So to be a little bit more give a little bit more detail on this, so that the general idea is really not new. It has been around in biochemistry and genetics since the 80s, and in evolutionary biology since the late 80s and 90s. And the basic idea is really that mutations that happen at structurally interacting positions should result in genetic interactions or epistasis. This is okay? Okay. And so just to give an example here, let's assume we have a salt bridge here on the side of the protein that's somehow important for the protein's functionality. Now, if we were to introduce individual mutants that reverse or flip the charge on these, this would destroy the salt bridge and therefore result in less functionality of the protein. And so our expectation for a double mutant would actually be that it's lost completely its function. However, if we introduce the double mutants, we've just swapped the charges completely, we might actually restore the salt bridge and therefore the functionality. And the differential between what we expect and what we observed is what we call epistasis. Now the simple idea is, if we were able to assay comprehensively these genetic interactions, we should be able to then predict which positions are actually close in space in the 3D structure, and use that to fold the protein. Right? So I'm going to exemplify you that, or the comprehensive mapping of double mutant activities first on this data set, which is kind of the gold standard demutational scanning data set in the field. It's on a rather short domain, protein GB1 domain, that binds immunoglobulin. And here the authors have performed a comprehensive large-scale mutagenesis, so they created all single amino acid mutations at all positions, as well as all double mutants, so all combinations of all single amino acid changes, which is in total more than 500,000 variants. And then they used a pooled in vitro mRNA display assay to determine a binding fitness. And this mRNA display is simply a clever way to link your genotype to the phenotype. You can then perform a binding assay, and by a deep sequencing readout, you can calculate how fit each particular variant is. So now we have a comprehensive assessment of the double mutant effects. Now we have to determine genetic interactions. How do we do this? So we can place each double mutant in this three-dimensional space where X and Y are the single mutant fitness values and Z is the double mutant fitness values. And now we simply classify each mutation as epistatic if it's more extreme than our expectation at these particular single mutant fitness values. And now, first off, we can just ask, given that we have this crystal structure from this domain, well, are epistatic mutant actually closer in space? And the answer is yes. They are enriched for being more closer, for example, at 8 angstrom cutoff. However, there's still the majority of epistatic double mutants is actually further away in space. It's quite far away, right? So now the question really becomes, how can we use this kind of information on a lot of epistatic double mutants to kind of prioritize which ones we want to trust to then actually fold the structure. So there's 2 scores I've developed to do this. One is simply asking, well, per position pair, how many of the possible 361 double mutants are actually epistatic? So it's really just doing the count statistics. And the second one is a little bit more advanced, and it's comparing how positions A and B interact with all other positions in the protein. And here what we're calculating is partial correlations on the kind of interaction profiles that you can calculate for each of these positions. And this is somewhat similar to the kind of unsupervised global statistical models that have been used in co-evolutionary structure analysis. Right? And now we simply combine these 2 scores to have a combined score. And this is what I'm going to present you now. So first off, we can ask, well, from this combined score, can we learn anything about secondary structures? And here the expectation is that in an alpha helix, a position in alpha helix should interact with its 3rd or 4th order neighbor due to the periodicity of the alpha helix, while in the beta sheet we expect that a position would interact with the 2nd over neighbor. And indeed, if we assay our score for these kind of local periodicities between close-by positions in the structure, we find that we can accurately detect the location and the extent of secondary structures in the GB1 domain. Right? If we conversely look at position pairs that are further away in the linear sequence, so non-local pairs, we find that our score is associated with the actual 3-dimensional distance in the protein structure. And we find that about 3/4 of the top positions are closer than 8 angstroms. And also we can look at this on a so-called contact map. We find that our predictions are actually kind of well spaced out, and where we're wrong, we're often only wrong by 1 or 2 positions. Right? So this is quite promising. And now can we actually use that to predict the structure? So only using the predictions we got from the deep mutational scanning data, so secondary structures and 3D contacts, we can actually simply put this into simulated annealing algorithms, standard algorithms that are also used in the co-evolutionary field, and we can accurately predict the fold of the protein down to 2 angstroms. All right. Now, does this work more generally? First off, we found that we can also use less complete and more noisy deep mutational scanning data sets. So this is 2 examples here of small domains where only subset of positions have been mutated, and from all these possible mutations, then only 10% of double mutants have been performed. Still, we find that we can accurately predict 3D contacts with about 60% accuracy. And moreover, we found that we can tap into the development that has been going on in the co-evolutionary field with deep learning, what we heard about in the morning, and basically do transfer learning. So we're using an off-the-shelf convolutional neural network called Deep Contact that has been trained on multiple sequence alignments of proteins with known structures. And we find that we can simply put on our predicted contact matrices, and they will be transformed to be much prettier and also have higher contact precision. And we find that we get about a 20% boost in contact accuracy from this. And this suggests that we should be able to do this in the future on shallower deep mutational scans, or noisier deep mutational scans, and that kind of all the developments that happen in the co-evolutionary field we can capitalize on. So this so far globular proteins. Can we also use this to ask other questions? And I'm just going to show you 2 examples now that we're really excited about. Especially because they're really hard to study with physical experimental methods. The first one is protein-protein interactions. Right, so this is a interaction between the proto-oncogenes FOS and JUN, a leucine zipper. And here we're looking at double mutants in trans. So for both of these proteins, all single mutants have been created, and they've been put together on a plasmid to create the double mutants. And the binding affinity of these double mutants can then simply be assayed by a protein complementation assay in yeast. So this is a classical assay where you have a split DHFR enzyme, and you attach your proteins of interest to it, and only if these protein variants, in this case, facilitate the interaction, then the two halves come together and yeast cells can grow. And so if we look at genetic interactions from this data, we find that we can accurately predict the contacts along the interaction surface with high accuracy. And moreover, if we apply our partial correlation score and look at interaction profiles, either across the columns, that it means between positions of Phos, or across the rows, that means across positions of Jun, we detect signals that correspond to the alpha helical structure of these proteins, suggesting that we can actually not only get the interaction contacts, but also learn something about the underlying structures of the interaction partners. And finally, second example is a new project we've recently put up on bioRxiv, where we were interested in learning something about disordered proteins. And so in this case, what we've done, we've deep mutational scan on the human RNA binding protein TDP43 that is known to phase separate and form aggregates. And it's implicated in the neurodegenerative disease ALS, right? And has a structured C-terminal region with RNA binding motifs and a N-terminal region and a C-terminal region that is prion-like, so disordered. And we mutated separately 2 chunks of this prion-like domain. And so we took all our mutant variants, about 60,000, of the human protein and transformed them into yeast. And then simply expressed the protein in yeast, the full-length protein, and assayed whether the variants are toxic to yeast. So this is our readout, toxicity. And now on this toxicity scores, we can also calculate genetic interactions. And what we find, so it's a long story, and this is just one particular aspect, but with relation to this talk, what we find is actually, we don't find any evidence for 3-dimensional structure in the genetic interactions, but we find evidence for secondary structures, and the 2 strongest signals we get for secondary structures, so here a beta strand and an alpha helix, actually coincide with structures that have previously been reported on short peptides in vitro experiments. Therefore suggesting that these structures actually form in vivo and are somehow related to the toxicity that the protein is causing in yeast. All right, and so with this I want to end. I hope I shown you that we have a genetic approach here to do what we call in vivo structural biology. And with this proof of concept, I've shown you that we can actually fold proteins from deep mutational scanning data. I like to provocatively say it's potentially easy, cheap in general, but whether this resonates obviously depends on the audience. So if you do computational structural prediction, obviously you wouldn't say this is easy or cheap, but if you compare it to established physical methods, you could argue it's potentially easy and cheap, especially if you come from a molecular biology or genomics background, and a lot of the labs in genomics or molecular biology have all the tools in hand to actually do these experiments and maybe start doing structural biology. In general, it is, on the one hand, because you don't need to purify or crystallize your proteins, but you also don't need a lot of homologs. So you don't have to have a big multiple sequence alignment, so you can potentially look at de novo generated proteins or evolutionary new proteins. Right? On the other hand, you need a selection assay, but there has been quite a bit of development recently going on to have generic selection assays that simply assay the stability of a protein. So these are potentially applicable to all kinds of classes of proteins. And, but most importantly, actually, selection assays do allow us to investigate structural conformations in vivo, as I've just shown you, for example, for disordered proteins. And this is something we're really interested about, and we hope we can learn much more about disordered or aggregation-prone proteins, or also transmembrane proteins, for example. And finally, more generally, I think the proof of concept that we can use genetic interactions to predict structures suggests that the inverse is true. That means that a structure is actually responsible for most of the genetic interactions we observed. And that suggests that we now might have an experimental paradigm to study the mechanisms of how protein structures and functions affect genetics and therefore also evolution. And hopefully we can therefore understand these kind of mechanisms better. All right, with this I want to end and thank Ben Lehner, our PI, as well as other people in the lab that have been working with me on these projects. Thanks a lot.
David?
Yes, I think it was a very interesting talk, and it was quite, the experimental stuff's very interesting. Obviously, I have a question on the deep learning side of it, obviously, because of the things. You use deep contact, I think you said that, is that right? That the deep contact for your, did you take care to remove that protein from the training set when you generated that figure, or is that just memorized, is that just recalled what it was actually trained on?
No, we didn't, as I said, we really used it off the shelf. So as it came when it was published by Jan Pang's lab, it was trained on the Astral data set. So we haven't excluded the possibility that, for example, GB1 has been in this training data set. However, we've seen this consistently now over many proteins, also protein-protein interactions, with obviously it hasn't seen before, that we get big improvements.
But that particular result is just memorization of the protein, essentially. I think it is.
I mean, quite generally, I think what especially here on contact maps is learned is the general patterns of alpha helices interacting or beta strands interacting. So I think most of the improvement comes from these kind of general patterns that we would observe in any kind of protein.
I'm just saying it is in natural force understanding kind of protein.
I was wondering if you could maybe comment on the importance of having the assay relate some biophysical parameter linearly to fitness. Because I could imagine another way you could get a genetic interaction is that everything in the protein behaves additively, but you have a nonlinear relationship between that biophysical parameter and the fitness function.
Yeah, that's obviously important that you find the right transformation that gives you basically the null expectation of no interactions. And we do this by using non-parametric fits to the data that kind of take out the global nonlinearities that you get, for example, from your actual selection assay, or the way you read out the sequencing counts later.
I have a question also. So now you do pairwise mutations, but I guess an alternative way could be sort of to random mutations. And so what is really the advantage of doing pairwise compared to doing some large-scale random evolutionary sort of?
So you mean random in the sense of randomly evolving proteins?
You don't have to evolve them, but you could really just have randomized codons on positions and a mix of that and don't read them well, and you will get some kind of sequencing afterwards.
This definitely also seems to be possible, and it's currently not quite clear which of the options are better, and this we'll have to see in the future. I think if you use pairwise mutations, it gives you a quite well-defined kind of framework in which you work and how you can interpret the signals that you actually observe.
But you did basically only one mutation in each, one amino acid replacement in each position, or?
Yeah, and all the combinations, so all the double mutants. Yeah.
But
you do all 19.
Alanines, or did you?
No, you do all 19
all
possible
19,
ones.
okay.
All possible ones. Yeah.
So we have 1 minute. Otherwise, we'll keep on. We'll have a little short break if no more questions. So, we have one here.
Actually, in your TDP43, were you saying you have no epistasis between any? So epistasis means it's getting less toxic, right? Is that right? No,
epistasis means you have unexpected effects, so non-independent effects.
Right. Yeah. But you read it as toxicity, right? You read it as toxicity ultimately.
Yes. But we find variants that are both more toxic and less toxic, right? So we have a dynamic range in both directions.
Right, but you don't find any epistasis between the helix and any disorder around it.
Not really systematically, so that we can conclude. We see some signal, but we couldn't really interpret it.
Okay.
Yeah.
Okay, so then it's time for next speaker, and next speaker